this black line right here. So, running along the course of the single-stranded DNA binding protein is the single-stranded DNA. And there are multiple DNA binding domains that are associated with RPA. So we'll just call this our single-stranded DNA in black, this black line. And there's multiple DNA binding domains. And again, basically, there's multiple DNA binding domains that are associated with the RPA, so it doesn't possess just a single DNA binding domain, but has multiple regions for this, and they're actually spread across the two subunits. And the DNA binding domains are all designated DBDs. So DBDs. Okay, and there's 3 of them in RPA 70. And so that's on the large subunit, and then there's a 4th one in RPA 32. And again, they're labeled in this figure as well. So you can see them there. Those are the DNA binding domains. And structurally, these DNA binding domains are essentially superimposable. They're very similar in their 3D structure. And the last thing that I want to point out is, and I don't think it's understood why, you kind of see the single-stranded DNA that's bound, that's traversing the protein complex in a very bizarre way. It's not just like you have a single-stranded DNA and protein that just sticks to it sort of linearly. It actually has this really complicated tract along the RPA molecule, and the reason for this is really not understood. So it has a complicated tract along the RPA molecule. It's presumed that, since if you look at these domains, you kind of have like little blob regions. Maybe these, little blob regions, there's some flexibility associated with the enzyme, so that these regions impart that. And so, the degree of dynamics that I guess I couldn't describe it as an enzyme really. I mean, the degree of dynamics that this DNA binding protein possesses is not really clear at this point, but it's sort of inferred from its structure, but reasonably has a complicated conformation. Okay, so that's our RPA, our single-stranded DNA binding protein. So now we've unwound our DNA and we protected the single-stranded DNA. If you present this to a DNA polymerase, you're out of luck, right? DNA polymerase really can't do anything with a template because it can't start DNA chains de novo. So it absolutely requires a primer that it can present, so that it can present a 3' hydroxyl for the polymerization reaction. And as I mentioned before, this primer can either be RNA or DNA. Okay, so again, I'll just draw this schematically. We'll need a 3' hydroxyl that can be presented to the polymerase. These little primers can either be DNA or RNA. Okay. And so what I want to introduce you now to is the enzyme that is responsible for producing primers in eukaryotic cells. And this is actually a protein complex with two different functionalities. So it has a DNA polymerase and also has a DNA primase. And each of them contain two subunits. Okay, so we have a DNA polymerase, and we have a DNA primase. And each of these contain two subunits. And so we have a total of 4 subunits that make up this complex. Okay, so the whole complex has 4, and this is basically depicted in A, really as a schematic of what it looks like. So the polymerase is referred to as simply polyalpha. So polyalpha, and that has 2 subunits. And then the primase is simply just referred to the primase. And again, that also has 2 subunits. Okay, and the complex, shockingly, is actually just called polyalpha primase. So there it's found, as a 4 subunit complex. These subunits function together, and they hang out, and they're best friends. Okay, so the polyalpha, if we can break this down, as I mentioned, has 2 subunits, and this is illustrated here in this sort of red color. It has a catalytic domain, and so this is where that catalytic domain is. So we have the 5' to 3' polymerase activity. And then there's kind of this, this extended C-terminal region, which interacts with sort of a structural subunit, and that structural subunit is labeled B here. That's this one here, this B subunit. And these things have molecular weights. The polymerase subunit is P180. Okay, and this second sort of structural subunit of the polymerase is called P86. Okay, so the C-terminal region interacts with P86. Currently, the functionality of P86 is actually unknown, but it's required for the activity of the polymerase. It's required for assembly of the 4 subunit complex. If anything, again, its function is still very unclear at this point. The primase, as I mentioned, also has 2 subunits. One is P58, which is this one here, and the other is P48, which is this one here. The P58 subunit is involved in binding the primase to the polymerase. So binds primase to polymerase. And so it's this sort of interface between the primase and polymerase. And then the P48 is the primase activity. So it has the primase activity. And the primase activity is simply a DNA-dependent RNA polymerase. Okay, so what this primerase does, it uses the DNA template to synthesize an RNA primer. Okay, and then what I've shown you on the slide again at the bottom here is a structure of Pol alpha primase that was derived by cryo-EM. So cryo-EM. And it's not really particularly of high resolution, but I think you can sort of see it. So it kind of looks like a bunch of blobs, and just to give you an idea of how the different subunits sort of relate to each other, over on this side where we have the blue, this is the catalytic portion right here of P180. So this is the blue portion, blue, and this is, you know, P86 up here, and again we don't really know the function of it, and it's hanging out at the top of the C-terminal domain, and then, you know, the primase is located in green, so the green is the primase. And you can see the P48 at the top. So that's the DNA-dependent polymerase, DNA-dependent RNA polymerase, and then P58 down here, which is making extensive contacts with P180. So kind of sort of bridging the small primase subunit to the polymerase. Okay, so that's the structure of DNA primase. Let me now show you a little bit about what this thing actually does. So continuing on with DNA primase. Okay, so you know how we have both have an RNA polymerase and a DNA polymerase all in one complex. So it's really not super clear the way the reaction goes, because really, you know, Pol alpha is like every other DNA polymerase. It really can't start a DNA chain de novo. So really, it's the primase subunit that has to do the initial priming. So primase starts by synthesizing a primer of about 11 to 14 nucleotides of RNA. So basically we'll have our primase, and it will synthesize about 11 to 14 nucleotides of RNA. So that's our priming, our initial priming event. And it really doesn't have a particularly strong bias as to where it will actually lay down the primer, except that the primer must always begin with an A or a G. So primer always begins with an A or a G. Okay, so what this primer looks like is a triphosphate A or G, and then anything from like 10 to 13 nucleotides. So we can rewrite this as an A or a G, where N is 10 to the 13 nucleotides. Another interesting thing about the primase is that it's slow. Okay, so it is slow. It only synthesizes about 0.2 bases per minute. So that's extremely slow. If we compare that to poly3, which we had talked about, was about 200 bases a second. So extremely slow. Pol alpha, by comparison, just make a note here, It's quite rapid, actually. It's about 1,000 bases per minute. And so the idea here is that once the primase subunit synthesizes the RNA primer, there's this rapid handoff to the DNA polymerase that then extends the RNA primer with DNA. And basically that's what's shown in this figure here. So basically we're creating an RNA DNA primer. Okay, so this portion would be laid down by the primase, and then this portion would be laid down by polyalpha. Okay, so that's by the DNA polymerase, and also this isn't very extensive as well. It extends it by about 10 to 20 nucleotides in length. So, you know, the product at the end of the polyalpha primase, which, you know, primes all of these DNA chains, is basically, you know, this very short, you know, RNA DNA primer. So it has an RNA moiety, and again, that's depicted here. That's extended by, or extended to DNA. And to give you a sense of how often this has to occur to replicate a human genome, you know, that's approximately, I guess, 30 million priming events. It's events in the human genome. And because, of course, every replication origin, you have to prime 2 leading strands, but then you have to prime every single Okazaki fragment. So, you know, the role of the polymerase alpha primase is extremely important. Curiously, given the importance of its role, Pol alpha does not have proofreading activity. So, Pol alpha has no proofreading. And so that's kind of wild, right? That you're making a substantial amount of DNA from a low fidelity primase. And, you know, like the expectation would be that this thing's fidelity would be about 10 to the minus 5, sort of like RNA polymerase. So the one thing that's inferred from this is that the DNA that's synthesized by polyalpha probably does not remain in the newly replicated genome. Once the Okazaki fragments are processed and joined together, you know, likely obviously the RNA moiety has to be removed. Like the replicated DNA doesn't contain RNA, right? Because that would be bad news. You know, it's really thought that this short bit piece of DNA that's synthesized by polyalpha is probably also removed before Okazaki fragments are joined together, and that this sort of low fidelity DNA doesn't likely persist in the final completely replicated form. So the low fidelity DNA likely doesn't persist. In the final DNA form. All right, so let's just summarize. We've unwound the DNA. We've stabilized the single-stranded DNA. We've primed it. We're more or less ready to go, except we still have a problem. And that problem is that the DNA polymerase on its own, even when presented with a prime template, it doesn't make DNA fast enough. And most of them have this tendency to just hop on and off the template. So the solution for this is actually kind of wild. And, to solve the problem so that your DNA polymerase doesn't hop on and off the template, is to provide it with a clamp to hold on to the DNA. And so this clamp, or sliding clamp, is a protein in eukaryotes that's called pCNA. So we call this sliding clamp in eukaryotes pCNA. And that's depicted as this pink structure in the figure. And you can also kind of see it right here. So this is looking at pCNA sort of from the, the top down. And you can notice that it kind of looks a bit like the helicases, right? I mean, it has a six-fold axis of symmetry. So a six-fold axis of symmetry. And it has this hole in the middle. And in this case, the hole definitely accommodates double-stranded DNA. So accommodates double-stranded DNA. And in contrast to the helicases, though, because it's not separating strands, it really doesn't use any energy. So it's not an ATPase. All it really has to do is encircle the DNA and bind to the polymerase to hold it onto the DNA. So basically, by looking at the structure, pCNA is a trimer. And the trimers are different colors here. So we have this green one, this blue one, and this yellow one here. And the subunits are identical. So identical subunits. And really is, its sole function is to promote processivity. Okay. So interestingly enough, these sliding clamps predominate and exist across evolution, and so that's what I'm showing down here in this table. And in the eukaryotic case, where the sliding clamp is pCNA, we can see that it actually promotes the activity and processivity of their DNA polymerases. But not every DNA polymerase can associate with pCNA. Especially in eukaryotes, it's only the poly delta and the poly epsilon. And the reasoning for this will actually become apparent in a few minutes. But basically, poly epsilon and poly delta are the enzymes responsible for replicating the leading and lagging strands. Poly alpha does not associate with the sliding clamp. So poly alpha does not associate with pCNA. And that's because you don't want this poly alpha hanging around and making a lot of DNA, right? Because it has no proofreading functionality. So polymerase alpha does not require a function in concert with the clamp. So you don't want it hanging around, making this low fidelity DNA. Okay, and if we look in E. coli, the sliding clamp is referred to as the beta clamp. Okay, so first what I want to do is actually show you a really neat little experiment that demonstrates what the sliding clamp can do to a polymerase. And this is what's actually shown here. So we have a plot that's off to the left. And it's basically a DNA polymerase assay. And the polymerase that's being tested is actually polymerase delta. And that's, again, a eukaryotic replicative polymerase. So eukaryotic replication polymerase. Okay, so it's a polymerase assay. Let's just review what we need to have in the assay in addition to our polymerase. So we're going to do a DNA polymerase assay. So we definitely need dNTPs for sure, right? What else do we need? So we need DNA, so we need a template. Okay. We just went through this. We need a primer. And, you know, we need some means of figuring out whether we've made DNA or not. So I think one good thing that we could do is probably put in some P32 labeled DTP. Okay. And then how are we going to visualize our synthesized DNA? So we could use Kornberg's method, and we could acid participate and measure acid versus acid insolubility, so soluble versus insoluble radioactivity. But this is a blot here, so it kind of looks like they didn't do that here. So another means we could actually do is actually use gel electrophoresis. And separate based on size. Okay. So on this plot here, basically what we would have would be the larger pieces of DNA that would be at the top. So the large, and as we can see from these molecular weight markers, and then, you know, the smaller pieces would be at the bottom. Okay, so we're ready to go. The first line here, number 1, actually contains all the components of the assay plus Pol delta. Okay, so here we have assay components. Plus Pol delta. Okay, and what do we see if you just have Pol delta present? I mean, what's the result? And again, the dark stuff that's shown on this plot is the radioactive material. So you can ask yourself, did we make any DNA? Well, we did. It's kind of small, right? Really, really down far on the gel. So we made a bunch of DNA. And when it was measured, it was approximately 184 picomoles. Okay, so that's very, very low molecular weight. So lanes 2 and 3, if we look at lanes 2 and 3, they're essentially the same. Both of these have, again, the assay components. So the assay components. Pol delta, and in this case for lanes 2 and 3 we have pCNA added in. Okay, so what happens when we add pCNA? And again, if you look down by the markers over on this side, you know, we ask ourselves, did we make DNA? And yes, we did. The next question is, did we make small DNA? No, because we don't see any down here. We actually made very large DNA. So the first thing that you can note is that pCNA results in this massive increase in processivity by polymerase delta, right? You know, if you compare lanes 2 and 3, you know, without the lanes 2 and 1, sorry, without the pCNA, you know, we're only making small fragments consistent with, you know, the polymerase hopping on and off, making a little and then dissociating. Whereas, you know, if we compare lanes 2 and 3, because we have the sliding clamp present, so this is with pCNA, the polymerase remains associated with the template, and now we can synthesize very long stretches of DNA without dissociating. Okay, so a couple things to take away from this is that the pCNA increases processivity of Pol delta. You can infer that the Pol delta remains associated with the template. From this because of these large fragments of DNA that we've made. The other thing, which I think is a little bit less apparent because of the way they loaded this gel, but there's a difference in the amount of the DNA polymerase that they added. So in lane 1, they had 184 picomoles synthesized, but they added a quantity of DNA polymerase that was only 51 units. And what the unit is isn't really important, but what I want you to focus on is that in lanes 2 and 3, where they made 51 picomoles of DNA, which on the face of it seems, oh, you're comparing 1 and number 3, they actually made less. But the key thing is they also use way less polymerase, so they only use 0.12 units. So if you do the math where you express how much DNA was synthesized per amount of DNA polymerase added in each case of lane 1, it's 3.6 picomoles per unit, and in the case of lane 3, it's actually 425 picomoles per unit. So in addition to this increase in processivity, the pCNA also causes a huge increase on the order of like 100-fold in the activity of the DNA polymerase. So the clamp doesn't simply hold the DNA polymerase onto the DNA to enhance its processivity. It actually improves the DNA polymerase's performance as an enzyme. Okay, so another key thing to take away from this experiment is the differences in the amount of polydelta added and the amount of DNA synthesized. Per polymerase concentration. And so we're comparing basically 3.6 versus 425, and that's almost, 100-fold increase in activity. Pol delta. Okay, so it not only holds on to the polymerase, it improves its performance. And really that's a theme that repeats itself throughout replication, in that you begin to understand that DNA is not replicated by individual enzymes, but rather it's replicated by a molecular machine that has components that function together vastly better as a whole than they do as separate components. Okay, so that's pCNA. And this is how the polymerases that replicate the chromosome are able to have sufficient activity and processivity to fulfill their functionality. So now we're presented with a problem in keeping with the clamp. We have a donut, and we have what's essentially an infinitely long piece of DNA in a eukaryotic chromosome, or in the case of E. coli, a circular piece of chromosome DNA. So the question is, how do we get the donut so that it's encircling the double-stranded DNA? So I see two choices here. We can either break the DNA or we can break the donut. Okay, so here's the question: how to get pCNA to encircle double-stranded DNA. So what are we going to do? We'll break the donut, right? And why would we break the donut? Well, we've been through this several times, and we all know that DNA breaks are bad, right? So we're not going to do that. Who cares about a donut? No one really, right? So we're going to break the donut as opposed to breaking the DNA. Okay, so we have to have some means of disrupting this donut such that we can load it to encircle double-stranded DNA where it can fulfill its functionality. So this is sort of performed by a protein complex that's referred to as a clamp loader. And the clamp loader in you and I is replication factor C, which was the third fraction that came out when Bruce Stillman's lab was doing some fractionation and reconstitution of replication reactions. And what is actually shown here in this top figure is replication factor C, okay, or RFC, which is the clamp loader. Okay, and it's showing its subunits, and it has a total of 5 of them, 1, 2, 3, 4, and 5, not necessarily in any particular order. It has 5 subunits. Okay, and in this structure, it's actually bound to pCNA. So here's pCNA here. And although you can't really see it because you're looking at the donut on the side, the donut is actually closed. So pCNA is closed. It's entirely intact. So this presumably would not be the form in which pCNA could be loaded. So this is just sort of displaying an interaction between the clamp loader and the clamp, and it's in its closed form. Okay, and in this structure, really, there are contacts between pCNA and RFC. And these contacts are between subunits RFC 1, 4, and 3, which makes contacts with pCNA in the closed form. Okay, and again, they would presumably be in this region right here. And what's displayed at the bottom of the schematic is where a piece of DNA is kind of sorted or threaded through the pCNA into the clamp loader. And what this illustrates really is that, the double-stranded DNA is going through the middle of the clamps. So this is our pCNA here. And it's also bound quite intimately in the middle of this, heteropentameric structure of RFC with the 3' hydroxyl right here, and the 5' hydroxyl kind of hanging out over here. So the point I want to make from this is that the pCNA RFC complex binds to what's called a primer template junction. And that's what it likes to bind to, which kind of makes a bit of sense, right? So, the pCNA RFC complex binds to the primer slash template junction. And basically what that would look like, if we just drew a schematic, so our 5' and this is our 3' hydroxyl, our 3', this would be our pCNA, and then we would have our DNA polymerase. this would be our pCNA. And that kind of makes a certain amount of sense, right? Because, this is where the position where ultimately we want our DNA polymerase. So ideally we would load our clamp onto the, this double-stranded region just behind where the polymerase is actually going to associate. So let's just delve into how this actually works once we open the closed form of pCNA. All right, so we've been talking about pCNA, and we need to solve the problem of getting, the pCNA onto the double-stranded DNA. So what is thought to happen, and this is largely from biochemical evidence, is that we start from a position where we have pCNA and RFC interacting just through 3 of the RFC subunits. Okay, so step 1 is just where we have 3 subunit interactions, and they are with RFC 1, 3, and 4. So that would be the closed form. If to that complex we add ATP, so we can add some ATP, we find that there is now much more extensive contacts and between pCNA, and that's what's demonstrated here, such that now in addition to contacting 1, 3, and 4, it's also contacted by RFC 2 and 5. As well. And what these additional contacts do is actually force the donut open, and you can see that here, kind of like a lock washer. So if it's starting out like this, now there is some discontinuity in the clamp through which the double-stranded DNA can pass. So this is the actual open form. Where we have RFC plus pCNA plus ATP. So now we're in a position where the RFC can actually bind to the primer template junction. And if you remember, I drew this on the last slide. We have our 3' hydroxyl here. We have our pCNA, and this would basically be our primer template junction. Okay, so now we have this discontinuity that's occurring, and so now basically the DNA can become encircled by the clamp. So we've got this thing going on where we've got this, this big disruption of the backbone. So, it's kind of obviously worthless because, the DNA can actually just, come out of this again. So what happens is that, the binding of the DNA promotes ATP hydrolysis by RFC. Okay, so DNA binding promotes ATP hydrolysis by RFC. Okay, and when ATP becomes hydrolyzed by RFC, the pCNA reverts back to its closed form. So then pCNA closes. And that means when it's closed, contacts from RFC 2 and 5 are gone. Okay. And the contacts with 1, 3, and 4 still remain, and pCNA goes from an open lock washer to a closed one. and now is encircling basically this double-stranded DNA. And then further downstream of ring closure, this then promotes the dissociation of RFC. Okay, so then you get downstream dissociation of RFC. Okay, so just to reiterate this process, we get, ATP hydrolysis, we get ring closure, and then we get RFC dissociation. And now we're left with this primed template. So we're left with primed, substrate, and it's really the perfect substrate for the polymerases. So we can add poly delta or poly epsilon. Okay, so the next step downstream of this would be loading of the polymerase onto the sliding clamp, at which point synthesis would ensue. Okay, so we've got our clamp onto the DNA, we broke the donut, mission accomplished. What I want to illustrate now is the roles of all three of these polymerases and eukaryotic cells that actually participate in replicating chromosomes. So as we mentioned, polymerase alpha, obviously a very important function, because it has to prime every single DNA chain. So if we look up here and we can identify this as the leading strand. We would actually initially have some poly-alpha primase priming, even the leading strand. But, once we load RFC and pCNA, which are depicted here, the polymerase that functions is actually called polymerase epsilon. Okay, and that's on the leading strand. So this is the one that actually does the continuous synthesis of the leading strand. So we have continuous synthesis of the leading strand. Okay, and it does so in the 5' 3' direction. 5' to 3' direction. Okay, so it's the replication polymerase, and as such, it has proofreading activity. Okay, so that's very important. As I mentioned, it associates with pCNA. And it is on the leading strand. And it's very high fidelity. And it has high activity and processivity. Now, if we look down here on the lagging strand, it's a little bit different because you have many Okazaki fragments, right, that are depicted here. And so Pol alpha primase has to be making many primers for every Okazaki fragment. And then after this short DNA extension by polymerase alpha, it is now substituted by a different polymerase, and that polymerase is DNA polymerase delta. Okay, and that's depicted here. And polymerase delta does the bulk of the DNA synthesis on the lagging strand. So synthesizes lagging strand. Okay, and in keeping with that, similar to epsilon, it has proofreading activity. And it's high fidelity as well. And associates again with pCNA, which is here. So associates with pCNA, and it is highly processive as well. Okay, so I mean, there's this nice specialization of DNA polymerase, of the polymerases. So one that does the priming, one does the leading strand, and one does the lagging strand. So you know, this then sort of raises the question: if you have this degree of specialization, how do you kind of get these things coordinated? And finally, the last thing that I want to leave you with is this amazing video, which I highly recommend that you watch once you finish the lecture. And it's sort of to illustrate what a replication fork looks like. And the first thing that I want you to take away from this video is that there's a lot of protein-protein contacts at the replication fork. So the idea is there is not like an individual enzyme that's functioning on its own, but rather this is a highly coordinated replication machine. As you'll see in the video, the DNA, the double-stranded helix comes in from one side, is unwound by the helix, the leading strand is synthesized continuously, and then we have the lagging strand where there's this cycle of clamp loading, polymerization, unloading, clamp loading, polymerization, etc. And it kind of sort of cycles through this so that, you know, each Okazaki fragment is synthesized, and it's synthesized sort of in the opposite polarity to the way the replication fork is moving. So you have this cycle of looping and release, and you'll see that clearly in the video. And so the point I want to make is that even though the polymerases on the leading and lagging strands are synthesizing in different directions, the whole process is very tightly coordinated. So you know, really have a look at this video so that you can visualize, if you're a visual learner, how all of these proteins function together in this highly coordinated fashion. And that's the end of replication.
These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present.